between D and A is 38%. So if I gave you those recombination rates, on a chromosome, you should actually be able to map out relatively where those genes are. So let's see if we can do this together.
So you said previous that the further apart the locations, the higher percentage of recombination occurs, right?
Yep, that's the basic assumption that we're going to work under. Okay, so according to those data, which two genes are located the farthest apart? A and B, right? Because they have the highest rate of recombination. Again, if it's a random process, rates of recombination should increase as two genes are separated from each other. So what we can say is, maybe A is located somewhere over here, and D is located somewhere over here. The question is, I'm sorry, that's B, right? The question is, there's a third gene, and who is it closer to? A or B? Why is it closer? Why is D closer to B? Yeah, because the rate of recombination is lower, right? So this between A and D has a higher rate of recombination than between B and D. So that means that D must be located like that. Is that cool? So if we were to draw out these probabilities, what we're saying is the probability of recombination between that and that is 24%. Recombination rate between this and this is 38%. And then between this and this, that's 62%. Right? Okay. So back to Hitchhiker's Thumb really quickly. Hitchhiker's Thumb really is a recessive character. So I have Hitchhiker's Thumb, and my wife has Hitchhiker's Thumb. What does that mean about our daughters? What proportion of our 4 daughters should have Hitchhiker's Thumb? All of them, right? Because this has got to be homozygous recessive. So the dominant character is normal. And the recessive character is Hitchhiker's Thumb. So if it's a recessive character, what's my genotype? Little h, right? So that's me. And my wife's has to be the same thing, right? Which means that all of our daughters should have Hitchhiker's Thumb. And you better believe I checked to make sure that each one of those girls.
Wait, so they do have it?
Yeah. You know, speaking of fingers, the crazy thing is, for the most part, right, the dominant character is going to be lots more common than the recessive character is. I think if we were to census the class, even though it's a small sample size, what we'd find out is relatively more of us have a normal thumb than have this bizarre Hitchhiker's Thumb, right? Same thing with eye color. The recessive eye color is blue. The dominant eye color is anything else. And I think that we would find that there are fewer blue-eyed individuals here than whatever else individuals. But that's not true for all genetic characters. There are some genetic characters for which the recessive phenotype is much more common. And a good example of that is finger number. So the dominant character for the number of fingers is 6. But the vast majority of us, right, all of us probably, have 5 fingers. And that 6-finger trait is like a true 6th finger. It has bone in it and it has musculature. That's the dominant character. But all of us, was anybody born with 6 fingers that's willing to admit that they're born with 6 fingers? all of us are the recessive phenotype. So even though it's recessive, it's much more common than the dominant character. I actually had a nephew that was born with 6 fingers. Neither of his parents had 6 fingers, but his 6th finger was a product of a mutation rather than an actual allelic inheritance. So my older brother, who's an interesting fellow, was playing with his baby's 6th finger while they were still in the hospital, and it just flicked off at his wife. So he flicked his baby's finger off at his wife. That's the kind of family I have. Another really interesting genetic character that's more common, the recessive phenotype is more common than the dominant phenotype, is a type of stature called achondroplasia, which is a type of dwarfism. The dominant character for us is dwarfism. The recessive character is normal stature, or what we define as normal stature. So if you're not a dwarf, demonstrating this particular type of dwarfism, you are homozygous recessive. So all of us in this class are homozygous recessive. Individuals that express this kind of dwarfism are actually heterozygous, because the homozygous dominant individual, that's a lethal combination, so those fetuses never actually develop. But the genetics look something like this, where for achondroplasia, this type of dwarfism, that's a dominant allele. And the recessive allele is normal stature. So the question is, what's the probability that 2 individuals that have normal stature will have a child with achondroplasia? It's recessive, normal stature is. So that means if we have normal stature, our genotype is it's homozygous recessive, right? So the probability that 2 individuals that have normal stature will have a child with achondroplasia is 0, right? But what's the probability that 2 individuals that have achondroplasia will have a child with normal stature? And that cross would be this. We know that the homozygous dominant individual is lethal, right? So those individuals won't develop, which means that if they're expressing achondroplasia, they have to be what kind of genotype? Heterozygous. Exactly right. So it's this cross, oops, and this, right? 2 heterozygous individuals. And that cross looks something like this. Right? So this individual's genotype is going to be what? Homozygous dominant. This one's going to be yeah. So we know that this combination is lethal, right? So this isn't even a possibility. So the question is, what's the probability of 2 individuals with achondroplasia having a child that has achondroplasia? And the answer is 2/3 or 66.666 repeating%. But it's also probable that 2 individuals with achondroplasia will have a child with normal stature, right? And that's a 33% chance. There was actually a reality TV show about that, right?
Yeah.
I never watched it, but Little People in a Big World or something like that.
There's like a bunch of, there's like little people in Atlanta and then like little people in like.
They expanded.
No, well, because I watched it one time and they, like, the lady had a baby that was normally sized, so she was like, she has to like.
Yeah.
Oh.
After
Water
too
all around with the
long,
baby.
the child is as big as she is. It's pretty crazy.
Interesting. So is it just because that our population kind of became more recessive over time, that it's more common to have a recessive trait versus
The answer has to be that natural selection favored this combination over these combinations. Yeah. Other questions? Okay. Okay, so a really interesting phenomenon that ultimately we need to talk about is this idea of sex-linked characters. There are some characters that are linked to either the X or the Y chromosome, and we'll figure out a little bit later in the lecture how we determine whether or not a character is sex-linked. But let's stay that conversation for just a few minutes. So, before we talk about some really interesting genetic anomalies, we need to remind ourselves about karyotypes and homologous pairing, right? So this is, bless you. So this is a karyotype of a diploid individual. So there are 23 pairs, kind of homologous chromosomes right here. And each one of these represents 2 chromosomes. So this is perhaps inherited from mom, dad. And each of those colors on those chromosomes indicate homologous gene regions. So is this individual male or female? If we look at this 23rd pair, it's male, right? Because it carries a Y chromosome. And in fact, biologically, that's how we define maleness, at least for our species. The presence of a Y chromosome. If there is the absence of a Y chromosome, and this distinction will make more sense in a few minutes, in the absence of a Y chromosome, the individual is considered female. So the vast majority of our chromosomes are called autosomes. They're just normal body chromosomes that control the production of somatic characters. But then we have a pair, at least in males, that's non-homologous, that are the sex chromosomes. So sex determination varies significantly between species. So in some types of grasshoppers, for example, the males have homologous sex chromosomes and the females don't, so it would be as if the females were XY and the males are XX. In most Hymenoptera, like bees, ants, and wasps, the females are diploid and the males are haploid, so the males just have half the number of chromosomes as females. They develop from unfertilized eggs. So sex determination varies significantly between species. But this is how we do it, right? If there's an X chromosome, or excuse me, if there's a Y chromosome, you're a male. If you lack a Y chromosome, you're a female. Okay. What's interesting is that sometimes malfunctions can occur in meiosis, where chromosomes don't segregate correctly. And that's called a non-disjunction event. A cell that ends up with an odd number of chromosomes is called aneuploidic. Or an aneuploidy is a cell with an odd number of chromosomes. A really good example of this is trisomy 21, right? Down syndrome. You have an extra copy of that 21st chromosome. That's aneuploidic. And that happened because at some point in meiosis, a non-disjunction event occurred. Chromosomes didn't separate correctly. Now, aneuploidic events can occur with autosomes, that is, these chromosomes that are not sex chromosomes, or they can be aneuploidic with respect to sex chromosomes. Now, the sex chromosome aneuploidies are really interesting, and so we'll give more examples of those than the autosomals. Generally speaking, autosomal non-disjunctions are lethal. The 21st autosomal non-disjunction is not lethal. It produces Down syndrome, but most of the others cause fetal abortment. Another really common non-disjunction event is the 23rd chromosome. I'm sorry, not 23rd, I keep saying that. That's the sex chromosomes, right? It's actually the 19th. But that almost always causes fetal abortment. And we know that trisomy 21, Down syndrome, is accompanied by a number of both physiological and cognitive disorders. So, for example, individuals with Down syndrome often have lots of respiratory problems at a young age. They have, there's a much higher occurrence of congenital heart failure in individuals with trisomy 21. Okay, But there are also non-disjunction events that occur within the sex chromosomes, and they are these four. These are the four that we're going to want to talk about. There is a type of non-disjunction where there are 3 X chromosomes. They're called metafemales. There is an XXY non-disjunction event, and that's called Klinefelter syndrome. These individuals are hermaphroditic. Male or female, first of all? Male, because there is a Y chromosome, right? So they will produce both female and male secondary sexual characteristics. So this individual would have like a penis, but it would also develop breasts during puberty. There is the XO non-disjunction event, male or female? Female, because there is no Y chromosome. Even though there's only a single X chromosome, still considered female. It's called Turner syndrome. There are often significant cognitive disorders associated with the XO genotype. And then there is the XYY genotype, and these are called metamales. Generally, there's no effect of having an extra Y or an extra X chromosome for males or for females. Generally speaking, these guys get a lot taller, like 7 feet tall, but cognitively and otherwise, they're normal individuals.
What is like this not scientific? What is the doing of expo?
What do you mean?
Like what do those women get?
So there are a number of cognitive disorders associated with it, but there are also some like physical symptoms. So for example, they can develop like kind of it's like their neck is a lot shorter because they have like a web of skin basically that goes from about mid-neck down to shoulders. They're typically reproductively not viable. They don't go through ovulation. They can have different facial structures. Generally the nose is a little bit flatter and the cheekbones are a little bit more subdued. Eyes aren't set back as far. Yeah. So the question is, who's at fault with these non-disjunction events? We know that it occurs within the sex chromosomes, but who's to fault, mom or dad? And if it's mom or dad, at what point in meiosis did that non-disjunction event occur? During meiosis 1 or meiosis 2?